going to get started. Everybody hear me? Good? Well, it's very exciting. Spent a lot of time, all the organizing. Not everyone's here, but you'll certainly see them throughout the conference. But yeah, it's just so exciting to see faces, all the names. So thank you so much for coming. And we're just so happy to be here and glad everyone can make it.
Hey everyone, my name is Miguel and I'm a PhD student here at Scripps. And I'm the co-lead organizer of AbGradCon this year with this beautiful team. It's been quite so Luke and I actually decided to apply to host AbGradCon in September. So it's been crazy 8 months, and most of all, we're very excited to have all of you here today. And that said, we're already on the search for the host for next AbGradCon, so AbGradCon 2024. So if you're interested in hosting next year, just come and chat with us in the next couple of days. We'll be able to give you an idea of what it entails. And without further ado, I want to introduce tonight's keynote speaker, Dr. Karen G. Lloyd, who is a subsurface microbiologist at the University of Tennessee. Although in 2024, she will be the Wrigley Professor of Earth Sciences at the University of Southern California. Dr. Lloyd discovers new types of life in Arctic permafrost, volcanic hot spring systems, and deep sea environments. She combines geochemistry with metabolomics, metatranscriptomics, metagenomics, and other environmental measurements to infer what life is like in natural complex systems. Let's welcome Dr. Karen Lloyd.
Thanks for having me, y'all. I went to AbGradCon here. What would that have been, like 2005, I think, as a attendee. So it's kind of nice to be back speaking. So I'm going to talk about life inside Earth's crust, but one of the reasons why we study life in Earth's crust is that the reason we're all here is that we want to find life outside of Earth. That is something that it's really kind of nice to give a talk here because I don't have to justify that. Like, we all agree. That is a thing worth doing. So when I think, where is life? So these are some good places, right? Like, I've named some nice planetary bodies that seem like a place you could find some stuff. But where in them do we expect to find some stuff? Well, the place we know the most about on these planetary bodies is the surface. And I'm not saying there's not life there. Definitely not. I mean, that would be ridiculous, but there's not like the Amazon rainforest sitting up there. We would have seen it if it was up there. So it doesn't seem to be like teeming on the surface, but the subsurface, if you think about the subsurfaces of all of these places, it's a pretty clement place to be. There's often liquid water, at least in the case of Europa and Enceladus, there's whole oceans worth of water. So that's cool. That's a major thing ticked off our list. Protection from UV damage. There's tons of stuff to build cells with. It's kind of a nice place to be. And the evidence for this is that, I don't know if you keep up with methane on Mars, but this has been something that's been kind of controversial over the years and now is no longer controversial. It is definitely there. We have methane in the Martian atmosphere. And the thing that I love the most about this methane is that it's not always there. We get whiffs of it. And so this is something that seems very lifelike to me. If, Titan is so full of methane. You're like, is that really biological? But for Mars, it seems kind of biological. So I wonder if right now there's a subsurface life ecosystem happening on Mars right now. It could be. And Europa's really easy, right? Like, this is this lovely artist rendering. If I had the artists that NASA does, I would get so much funding. But they've got this ice cap and then these occasional jets. Enceladus definitely has jets. And absolutely, for 100% sure, there's a giant ocean of liquid, probably saline water on Europa, which is just very intriguing. Okay, so a high probability shot for astrobiology is to look for life in the subsurface of planetary bodies. So we've got this planet to look for. So can we use Earth life, the life we find here, as a guide for what might be there on the subsurface of these other planetary bodies? And I don't know if you guys ever do this view of Earth. Do you ever try to get the land out? It's like Hawaii, and New Zealand, California's fighting them to gain place. But I love looking at Earth like this because it really reminds you that we are an ocean planet. We know that, but you can look at it from the backside. And some of my colleagues have added up the total number of microbial cells that are living underneath the ocean, in the subsurface, in the marine sediments. And it's literally astronomical. It's 10 to the 29, which is a third of the microbes on the planet are buried in the subsurface underneath our oceans, which is 10,000 times more than the number of stars in the universe, although that number has a big error bar on it too. So actually, most of the life on Earth is actually inside Earth. So we are a subsurface planet too. And if you look at the terrestrial environment, this has pretty much the same amount of microbes in it. So it's not just the oceans. This stuff is kind of everywhere. Okay, but if you think about microbes and life inside Earth's crust, our dominant signal is things like the Amazon rainforest. I mean, there's no question that this planet has tons of surface life. So you have to ask the question, is it real? What are all those little creepy crawlies? Are they meant to be there, or are they just like the leftovers? It can be dangerous to assume that an organism is optimized to live just where you found it. I have this really self-deprecating anecdote that hopefully you guys will get. So I did the geobiology, the Agron geobiology summer course, which is now at Penn State. Do you guys know about this? It's fantastic. You should look into it if you don't know about it. It's I think a 6-week summer course, and this year it's going to be in Italy. Is anybody going? No? Okay. It's going to be great. Jen McElady's running it. It's Penn State's first year, so maybe it's a rocky first year. Go next year. But I did that in the second year that it was ever around, and we went to Yellowstone for our field trip, and I was just like so full of wonder, and like my eyes were saucers, and I was taking in everything that I could, and I was watching in this hot spring, I was watching a spider in a hot spring, and I was like, oh my God, this place is magic. Even the spiders are adapted to high temperature, and I was just watching this spider just like, cool, just like chilling in this hot spring, just thinking, oh my God, I need to work on arachnids. Like, I got to switch my whole thing. And you know where this story's going, right? It died. I watched the thing die. It just like curled up, and then I was like, oh my God, Karen, you're such an idiot, But that we have to be conscious of that. Like, are we just looking at something that's kind of not really supposed to be there? Well, I would like to convince you in this talk that that is not true for the subsurface. That it's got a lot more going for it than a spider in a hot spring. So here's my list of evidence that the deep biosphere on Earth is real, alive, and not just leftovers from the surface. First of all, there's a lot of energy sources in the subsurface. It's not just like marine sediments that are sort of piling up. So I see this figure a lot, but it's nice because it encapsulates a lot of different environments. So we've got polar regions underneath glaciers. We have deep anoxic brines. We have hydrothermal vents. We, of course, have marine sediments, which cover so much of the planet. We have subduction zones. We have serpentinizing environments. We have volcanic adjacent areas. And then we have human-made areas as well for human-made environments. But all of these things are places where you can find nutrients to drive a subsurface biosphere. So that's one thing it's got going for it. The other thing is that when we look at what's down there, we actually see evidence that these things are breathing, that they're alive, because we can see the chemical signature that they leave. And if you are a marine sediment person like I am, you live and breathe these kind of diagrams. But if you're not, if you look at small, faint things next to bright things in the atmosphere, then maybe you're not used to looking at stuff like this. But this is my life. This is up here, that's the ocean. Up there, this is the surface of the mud, and we're just like looking down into the muck. And look at the depth scale on this. This is meters. So this is like drilling a football field worth down. So this is a massive drilling operation. And then if you section this thing into sections, and you measure the concentrations of, in this case, sulfate as you go down, and then methane as well. The fact that this is curved like this, and we can measure the sedimentation rate, we can use those two factors and then just what we know about diffusion and compaction and basic things like that to calculate the rate at which these organisms are breathing the sulfate. And the fact that it's got this concave shape means that somebody was breathing it. You can't really get that shape unless you have upward advection of sulfate-free fluids, which we don't have in this situation. So this is evidence that these things are breathing and they're alive. And it's not like the stuff we find down there is just surface stuff that's hanging on. It's its own thing. So if you do, this is a easiest way to show this. This is one of my colleagues' papers. This is a NMDS, what does that stand for? Non-multidimensional scaling parametric test. This is a axis-less axes, but all you have to know to understand these data are that dots that are close together have more similar microbial communities, and dots that are farther apart have more different microbial communities. So all the seawater grouped together, and all the marine sediment grouped together. And anybody who works in seawater and sediment just knows this intuitively. We have almost no overlap in the types of microbes we find in these places. So it's its own thing. And then the last thing is much harder to show. And I've spent a lot of my career, and my students in my lab have worked on this. How do you show they're actually adapted to live in this place? That's been a little more, I'll spend a little bit more time talking about this because it's a bit more nuanced. But I'll first not go into the whole story, but just talk about one of my graduate students, Jordan Bird, took samples from an IODP expedition on the great ship Manisha, not the Joyous Resolution, which went to the Baltic Sea and drilled hundreds of meters into the Baltic Sea. And he combined metatranscriptomes, metabolomes. If you don't work with those things, we basically, if you think about like a cell as a bag of biomolecules like DNA, RNA, proteins, lipids, and all that kind of stuff, and then all the chemicals that get pushed along by those processes, if you take a natural sample and you pull all those chemicals out and then just say, well, what was there? Then you can kind of back calculate what that cell was doing. You can reconstruct how these organisms were actually functioning, and that's the kind of stuff we do. And so from that, we found that it looks like they do have some adaptations to being there. So we can see some actual chemical evidence, not just in genes. So remember, this is why I'm sort of harping on this, is that you can predict a lot of things from genes, but we saw actual evidence that these functions were happening. We saw a DNA repair that consumes NAD, which is a cofactor that gets redox cycled in a cell, but in this case is getting used for DNA repair. We found an accumulation of osmoprotectants, so this is not a particularly saline place. We kind of think that collecting things like trehalose inside cells is a lot about shutting down and slowing down the cells, like pickling themselves. And we were able to find, we discovered ecology, which maybe doesn't sound like such an exciting thing to find. But when you're dealing with an ecosystem that might kind of be dead, finding ecological niches is a great little indicator that they're actually alive. So we found that microbe A eats food A, and microbe B eats food B. And we showed that with functional assays, which still blows my mind that that actually worked. So this may be something that they do to not fight, because basically, when you don't have a lot of resources and nobody gets to come in and nobody gets to leave, if you compete, then it's mutually assured destruction. If you kill that cell, it's never getting another one. And then we found this one group, if you are a microbiologist, this might make sense to you, the Atrobacteria, which is a deeply branching uncultured clade. We think it eats this thing called allantoin, which is a nitrogen-rich degradation product. And we think it's maybe a keystone of that community, because it looks like, of all the transcripts, so again, those are the mRNA that are going to turn into proteins, that were the most highly transcribed in this whole 8,000-year-old sediment, like 50 meters down into the seafloor. The number one most transcribed gene, which if you've worked with transcriptomes, you could probably guess what the number one most transcribed gene was. Hypothetical. It's always hypothetical. It's like those things are like the most important genes in the whole ecosystem, and we have no fucking clue what they are. So the second thing that was most transcribed was actually a sharing gene. So this is a starving environment where they don't have much to eat, and the second most highly transcribed gene was actually a transporter that's used in centrifuges to share your food with others, which is very touching. But it also, basically feeds into one of my sort of ongoing theories that these extreme apocalyptic ecosystems are not dog-eat-dog at all, because it doesn't make sense to compete when things are so limited. They actually cooperate, I think, quite a bit. This is another little piece of evidence that we have some adaptation to this environment is work that came from another one of my PhD students, Katie Sipes, who is so absolutely addicted to going to the Arctic after going. She can't stay out of it. It's so funny. She keeps finding more projects, and I'm like, Katie, where are you? And she's like, Greenland. So what she found in Siberia, this is this beautiful Kolyma River. This is some of the oldest permafrost on Earth. So this has been continuously frozen for 1 million years, which is crazy. She found this group, which I was telling some folks at dinner about, this Thermoprofundales group is only ever found in marine environments. And she found it in this freshwater place. And a lot of these clades that she found in this deep, weird, old permafrost are very similar to what we find in these other subsurface environments. So it really seems like there's something going on. And the last sort of adaptational story I'll tell is from another one of my former students, Joy Bongiorno, who looked at these transcriptional differences. She has this amazing data set, which actually we're still analyzing. But depth is here. This is now centimeters, so this is like over that much space. This is not super deep. But what she looked at was the total amount of transcription of each of these genes as you go with depth. And this is the amount of transcription on the y-axis, and you can see it's in log here. So this is quite a huge increase. So she found this very big ramping up of this transcription of this one gene, which ended up just being a gene to basically turn it into a spore. It was like a sort of like a strange type of sporulation. So we think that these things, we think that there are adaptations to being in the subsurface, you know, sporulating or repairing. Basically, we're not done. I can't say that definitively, but all of my research sort of points at the fact that these things are actually adapted to be where they are. So now, maybe I've convinced you that Earth does have a vibrant and varied subsurface biosphere that's supposed to be there. It's not just waiting to get to the surface again. It means to be there. So how can we use this? How can we use this to look for life on Europa? So I think of the subsurface as being divided up into two main categories. One is subsurface life that gets its chemicals from inside Earth. So it's basically got its own lunch with it, and it's making its own biomass. So chemosynthesis, that's making biomass from chemicals. And then the second type would be those that are dependent on photosynthesis, which are sort of like dependent on the surface world to give them food and to feed them. And the second one maybe, I mean, it's like, it doesn't seem as exciting, right? If we were talking about subsurface life, you kind of want it to have its own thing. And like just waiting for stuff to trickle down from the surface seems like sort of cheating or maybe not what we're going for here. But I wanna argue that this second group is actually useful for astrobiology. We can still work on these places on Earth that are dependent on the surface and learn some stuff from it. And I also forgot to say, since it's the evening and we have a lot of different area of expertise, if you want to ask a question, we're not going to ask too many questions. It's for technical reasons that you guys can't ask a lot of questions right now because there's a microphone. But if you're lost, just raise your hand and be like, can you explain this one thing? Okay, so anyway, forgot to say that at the beginning. So is this second one useless for astrobiology? Do we not want to study places like marine sediments because it's dependent on having Amazons up at the top? No, I think it's useful because we can get to them. Which, I mean, I know that's not the best reason to study something, but it is important. Like getting a sample is very important. And the cool thing about marine sediments is that they're layered. So if you want to run an experiment for 1 million years and I want to run an experiment for, I cannot tell you how badly I want to run an experiment for 1 million years, but obviously I cannot. But marine sediments have already done it. And so if you can be creative about how you sample them, you can test hypotheses that have been run for 1 million years already. That's why they're magical to me. So I want to tell you about the kind of life we find in them by talking about this first great expedition that we had off the coast of Peru with the ocean drilling program. It was not the Integrated Ocean Discovery Program back then. It was ODP. And it was the first sort of aseptic samples that we got from the deep subsurface. And I'll show you what we got with the simplistic, not naming the microbes, but I'll show them with boxes. So the dark blue boxes will be a strain of microbe that somebody else discovered, and the gradient to white will tell you how far away it is from something somebody's discovered. So like the novelty of it. If it's totally novel, totally new phylum that's never been discovered before, it'll be a white box, and then shades of blue in between. So what we caught with DNA sequences was a bunch of kind of light-colored boxes. This stuff was really new. It was very much unlike anything we'd ever seen before. This is what I did probably almost exactly at where most of you guys are in your careers right now. I was pulling these sequences out and using far less sophisticated bioinformatics at the time to figure out how closely related they were. But it was, we didn't know. We didn't know about this uncultured dominance of life like you guys do now. We were finding it. We were like, what the hell is this stuff? I don't know. So obviously, the first time you go to a place like this, you want to start culturing stuff, and this is low-hanging fruit. It's full of new stuff. We should be able to grow it. So I'll show you the results of the growth experiments, and every time they got one of these guys, I'll show you the new one underneath it, and then any extra ones I'll show you off to the side. So these are the ones of these that they caught when they cultured. Do you see any overlap? Oh. Do you see any white boxes? Got the same thing that people always get. And this is not to say that we shouldn't culture. I really don't want to send that message at all. That is the wrong message. And there's a lot of novelty, even in here, just getting something that looks like something we've seen before out of the deep ocean is absolutely worthwhile. But we were not catching these guys. And there actually was another group that did this too, and they got pretty much the same thing. So this really hit home to me, and is one reason why I really stick to this biomolecule stuff. We often call this stuff, have you guys, I'm sure you've heard this term, microbiology. This is controversial. Does anybody hate that term? No. Good. Accepting. Maybe the astronomers don't like it. Yeah? It's like stealing it. I actually like this term because it signifies just how pervasive this stuff is. It's just kind of like, oh, it's not like, oh, we found a new thing. It's like, this is the main thing, and we don't know what it is. But yeah, in terms of total mass, it is far, far less than astronomical dark matter. So since then, we did a study where we went through and pulled out gene sequences from the subsurface and asked the same question for all of them. Like, if we divide up, like, how many of the sequences that we find are closely related to a culture versus something else new entirely, and then we sort of put them in categories, we find that this biggest pie wedge from terrestrial subsurface metagenomes is novel genera to class. And then we get almost more than a third are novel phyla. So they're really dominant in these environments. That's the bacteria, and that's the archaea. So it's not just that they're there and they're dominating diversity. They're actually dominating the total biomass in these subsurface environments. They're totally new stuff. And my other soapbox about this is that even, you can look at this little yellow pie wedge. So this is a cultured organism. This is done. The things that fall into this pie wedge are things that you can order from a company and grow in your lab. However, even these ones are probably going to act differently when we study them in the subsurface than when they grow in our labs. I mean, if you were like a lion researcher, which would be awesome, but if you studied lions only in a zoo, then you would be like, lions, their whole deal is that they eat little prepackaged things of meat in cellophane, and that would be your conclusion. I mean, that's stupid, right? We know they hunt and they kill live things out in nature, but this is kind of like studying microbes in a zoo. So even for the cultured guys, we still need to go out into nature, and we still need to sample them where they are and try to figure out how they live. So, soapbox number, I don't know what number I'm on now, many. If you take Microbiology 101, which I now teach, we call it 220 actually, but like your basic microbiology class, microbes grow in cultures. That's how we learn about microbes. And we know a lot about what microbes do on this planet. These are like basic truths. I'm not saying they're not true, but I'm saying this is a frame of reference. I want all of you to not think of these things as necessarily true. Just, life could be different than what we think. And it could be that there are microbes that are never going to grow in culture and are not going to act like anything that we've seen before. And we need to not limit ourselves, especially as astrobiologists. You know, we can't think small-minded about this stuff because we'll miss something really important. So how fast do these things grow in these sediments? That's this is part of the taking the lid off and seeing your implicit frame of reference that you may not have even realized you have, is when we think of microbes, maybe we think fast because pathogens grow fast, right? You get sick pretty quickly. E. coli, for instance, which we've all worked with, certainly has a doubling time of 30 minutes. It's not even the fastest microbe, but it's pretty fast. But because we can work in marine sediments, we can do time scales because we have these layers. So we can actually measure the doubling time. So if you look at sediments with these ages ranging from 100 years down to 5,000 years, and you put the generation time in years now, that was 30 minutes, this is years. And at the surface, in 100-year-old sediments, it's still 0.1 year. So this is like still very, very slow compared to E. coli. But if you go down even further, we get up to the tens of years and maybe 100 years generation time, which, you know, we publish papers with these numbers in them all the time, and we throw them around all the time, but it's hard to wrap your brain around what it's like for a cell to actually live for that long. Because this is a generation time. This isn't necessarily dividing. This could be just replacing itself, like a turnover, total carbon turnover for these cells. So you know, the number of cumulative generations doesn't really increase at some point. So what we have are a bunch of organisms that seem to just be hanging out for how long can an individual live? Does somebody want to venture a guess? One, I don't know, nematode, tardigrade. Does anybody have a feel? Like, what do you think of when you think of a lifespan? It's the longest lived thing. You guys are all astrobiologists. You'd be like, forever. Yeah.
I don't know, thousands?
Thousands, yeah.
1,000, 5,000?
Yeah, I mean, even saying like thousands or 5,000 years for a lifespan for one thing is already like outre, right? That is really long. Like we have sequoias are what, 300? Bristlecone pines are maybe 1,000. Do you have a thought? Aspens.
Yeah, because they have the clone type.
Oh, yeah. How long do aspens live?
The longest living one was like 10 million or something. It's like ridiculous.
10 million. How about,
But it's like weird. It's not
does that, but even, right, and so those guys, so it's like clonal, right? And then even like the sequoia that's not clonal, it still has that living tissue. And the living tissue certainly, like those cells, a living cell doesn't last as long as the whole tree. So yeah, we've got things that what I'm saying are lasting for 100,000 years. One cell, one like little precious tiny little lipid sac is hanging out intact for 100,000 years. And I'm going to venture to say also 1 million, because why not? And I don't know, man. Like, it's crazy, but they might. Extra evidence for this, because it's crazy, is if you add up the total power. So remember back to physics, power is energy over time. It's the flow rate of how much energy you can feed something, because you can have a very nice quantum of energy. Like a pizza will be just fine for me for a day, but a pizza is not going to last me for my lifetime. I'll die. So the delivery rate of your food really matters. So if you add up the total power per cell, because we know pretty much how many cells there are out in marine sediments, this right here is the lowest measured power per cell that somebody has measured in a non-growing chemostat. Have you heard of retentostats? I really love retentostats, but people don't work in them. A chemostat is a sort of normal thing to do where you're passing fluids through a population and you have them grow at a certain rate and you're constantly feeding them and growing them. A retentostat just has a filter at the end. So nothing is leaving, so they're not regrowing, so you just keep them there. So in a retentostat, they get down to this extraordinarily low power, but then if you integrate the total power available from sulfate reduction and from methanogenesis, all underneath the world's oceans, it is orders of magnitude less. than what it takes to just keep a cell going, not growing. Growing's like way out here. Growing's not even on this chart. This is just like existing. So it's weird, man. It's real weird. And this is, the reason I'm emphasizing this to you guys is that this means that there's basically a lot more energetic space for life in this universe than we think about in our daily lives. We can imagine more for where life exists than, just, my dog and cat and my kids and stuff. What we've learned from marine sediments is that life in the subsurface may be fundamentally different than what we've studied before. And we can survive on much less power than we experience in the surface world. And this 100-year lifespan thing, which we're kind of intrinsically stuck with in our brains, is an assumption. It's not a requirement. Okay, so I want to switch gears. How far in this? I'm 37 minutes into this. Well, no, it's not quite 37 minutes. I guess the bar's closed. I can't tell you guys to get a drink. Does anybody need a... I've got a drink. Does anybody have a question before I go to the second section of this? Yeah. Oh.
So there's a lot of chloroflexy, right? Do you think that those are also adapted to the subsurface? And what's going on with why but they photosynthesize? Why?
Oh, that's a good question. So
they
for those of you who didn't hear that because the microphone wasn't there.
Oh, sorry.
Why are there so many chloroflexi there? Are they adapted to the subsurface? Because they are photosynthesizers, or at least phototrophic. They are not all phototrophic. Yeah.
Oh.
So Dihalococoides is a genus within, there's a lot, but the dehalogenators, I guess you could call them. So that's where a lot of the chloroflexi research has been done, just because that's funded, because it's bioremediation, because we use chlorinated compounds to clean planes and stuff. So those are fascinating organisms in and of themselves. But most of the chloroflexi that we find are Anoralinae. I don't know if you know that group. It's not well cultured. We don't have any phototrophic genes in them. But yeah, there's a lot. It's cool that you bring up the chloroflexi because they are one of the most mysterious phyla. Do you work on chloroflexi?
I'm working with steam vents, and I'm in Jimmy's Law Scripps, so we do a lot of thermophiles and stuff. So chloroflexi keep popping up, and there's a lot of them, and
Yeah.
I just keep looking at the metagenomes, and I'm like, yeah.
So the study that we did where we quantified the uncultured things all around the world, we looked at all environments, the only clade that showed up in the most uncultured, that showed up as one of the most abundant clades in every environment, animal guts, engineered places, human guts, snow, everything, was Anoralinae. It's that chloroflexi group. Everything else shifts, and you have some things in some places and none. But Anoralinae, I don't know if that's how you pronounce it, they're everywhere. They drive me nuts. I don't know what they do. I guess microphone javelin's a bad idea.
Do you think it's just the doubling time, or is it nutrients and potentially physical structure that's a problem for culturing microbes in the sediments?
Oh, why don't we get cultures?
Yeah.
I wish I knew that. I think it's all those things. It's if you think about what culturing is, it's limitless access to abundant substrate, which is a problem if you don't have, running your metabolism really hot is actually dangerous for a cell, and those of us who run at a very high level have all kinds of reactive oxygen species defenses and things like that. But if you're not set up to go fast, then being forced to go fast can kill you. So that's one thing. Too much food. You need your friends. You need your enemies. You need vitamins. You need minerals that you're lacking. You need them in the right crystalline structure. Pressure. I don't know if it's self-congratulatory to talk about my TED Talk, but I gave this TED Talk about whatever, it happened. I gave this TED Talk about culturing things in the deep sea, and I get an email, probably, it's slowed down now, but it was hot and heavy when the first thing first came out, somebody being like, have you considered pressure? Yes, I've considered pressure. It's I forgot to mention it in the talk, but people are thinking it's good. There's a lot of reasons why things are not on culture, but I think there's a lot to learn from that.
Hypothetical, and I guess philosophical question. Do you think a cell could possibly never die? Or is it possible that the lifespan could just be essentially infinite?
We have a limiter on that, and it's 4.5 billion years. Yeah. But that's it. I mean, but that's that's a lot to play with. And then if you invoke panspermia, which I don't know if you know this theory that life formed outside of, which I wanted to switch to panovia, but then I don't know, whatever, panmovia of life, then you could have a living cell that was like when the Noachian period on Mars was, you probably know better than me, like 5 billion years ago, 6, somebody knows. Anyway, Mars is older than Earth, so that may be our limiter. I don't know, but that's crazy. I'm not that crazy.
So you mentioned about the abundance of syntrophy in the deep subsurface communities. Do you think, are there any interesting patterns in the distribution of microbes that are typically associated with syntrophy or kind of syntrophic communities? Yeah. Or is it always just nutrient limitation?
You mean physically where cells sit relative to each other? Do they sit together? Different layers within the subsurface. Yeah, do we see more syntrophies in some areas than others? I don't think we have that kind of knowledge, that resolution. You could maybe do that project, but I don't think we have that. Just concluding syntrophy from the transcription of some genes is already kind of sketch. So, but you could push it farther for sure.
Okay. Hi, I really like your talk so far. So I have done research in the deep subsurface as well. And so you had a slide that showed the different adaptations for the subsurface microbes. And I think you mentioned that you had transcriptomic data, genomic data, metabolomic data. Yeah. So it's a lot of data. Mm-hmm. And I only work with genomic data, and already I'm so overwhelmed by the different things that I can analyze. So I was just curious. I mean, I could read the paper, but you're here now. How do you find the adaptations in this mess of data? Yeah. And how does it compare? So you mentioned DNA repair mechanisms and all of this. How does that compare to the terrestrial counterparts? Mm-hmm.
Yeah, those are really good questions. I mean, I think this is something that we all face. There's in every project, and I've seen it happen to all my students, you work so hard to get that giant data set, and then you get it and you're like, what do I do with this fire hose of information? And it's not how you analyze it. There's no prescribed path through it, which I think is a wonderful thing, because it means somebody could take your same data set and draw completely different conclusions from it. But in the case of the stuff I was talking about, we started with our smallest data set and our most valuable. So we did metabolites, which, if you haven't worked with these, this is you do liquid chromatography coupled to mass spectrometry, coupled to another mass spectrometry to sort of spread things out and get monomers. So you can basically get things at the amino acid, monomeric amino acid level, and you kind of get what you get. And so I only did it because my buddy does this down the hall, and he was like, let me put some of your weird samples in the machine. And I was like, that's insane, because you're just going to get the organic matter that's around. This stuff is not necessarily connected to the living microbes. And he's like, let's just try it. I was like, okay, sure. And he was like, it totally failed. It didn't work. And I was like, well, yeah, nothing ever works in the subsurface. It doesn't surprise me. But he was like, I only got 20 metabolites. And I was like, 20's a lot. So we took our 20 and just backed off of them. We said, okay, we've got nicotinate. And I was like, what in the world is, I Googled it. I was like, what is this and what is it used for? And I was like, oh, it's a precursor to NAD. And then we also had quinolinate. I was like, never heard of that that compound before. And it's also an intermediate in the pathway for the creation of NAD. And so then, because we had the transcripts, we were able to say who is producing the biosynthetic pathways for these molecules that we see. And so that was a nice way to do a data reduction. Those may not be the most important stories in those ecosystems. In fact, they probably are not, right? We probably missed the biggest story that's there. But that was just the one we found. I don't know if that's helpful.
That's super helpful. Yeah. Metabolomics are great.
They can be great. They can be confusing too. But it's also when you're dealing with a gigantic data set, I guess just don't worry about getting the perfect story. If you can find that it says something, run with it and publish it so we can know about it too.
Thank you.
Hi, thank you for your talk. You talk about these cells that live for hundreds of thousands of years, or like millions of years, or billions of years. But maybe philosophically speaking, what do you consider to be an individual? Like when all the biomass kind of regenerates or whatever, recycles. Thank you.
Is it the same?
Is that still the same individual?
Yeah.
Like, how do you work around that?
Yeah, I don't know. I mean, I guess that is a truly philosophical question. There's like the philosophical point about like my grandfather's axe. Do you know about my grandfather's axe? There's something in like, this is my grandfather's axe. It's, you know, he had it, and it's like, well, I replaced the blade a little while ago. Well, I replaced the handle a little while ago. I replaced the leather strapping on it. It's like, okay, no molecule of that axe was ever held by your grandfather, but it's my grandfather's axe. So then that's a purely philosophical question. But I think there's a second philosophical question, which I find interesting, which is what is an individual, which is what you said first, which is like, when we talk about microbes, we tend to talk about the individual as a collection of cells. We talk about a strain. You know, we don't talk about a cell. Like an individual cell is pointless in real normal microbiology because they're constantly regenerating and we have cell lines and stuff. But in this environment where you can't have a cell line, suddenly the individual takes on great importance. This is almost akin to like our lives. Like we care about an individual dying, and we don't care about a cell of a microbe dying ever. Makes research a lot easier. Okay. Should I continue? Okay. If you're bored, take a nap. It's warm in here. I gave a talk once on a ship and everybody fell asleep. It actually happened. It was nice. They looked very comfortable, so I just kept talking. So now that we know that we've got all this weird stuff down there, how can you sustain a subsurface biosphere? We know that there aren't a bunch of plants at the surface of these other planetary bodies, but can we work on this planet where we are blessed with all these wonderful plants up here and find things that are independent of that dominant signal? And so this isn't the only way to go about this. Hopefully you're a room full of people who will find other ways to answer this question too. But one way that my colleagues and I thought of doing this is to go to a subduction zone, which are fascinating geological places because in a case of an ocean continental subduction zone, you have an oceanic tectonic plate that basically just gets shoved very slowly into a continental tectonic plate. An oceanic plate is just denser than continental plate, so it sinks and it goes down. And this is one of the major ways that we get recycling between the deep Earth and the surface Earth. But what also happens is that you get this buildup, this bunching up in the forearc, so this is the ocean plate going down, that pushes water out of all these areas. And if you draw this 150 degrees C isotherm, which is kind of the limit of life, although I definitely won't say that because it could be much higher, but currently, you know, we think that life has to be under this value. There's actually a lot of real estate in here that's probably getting flushed out passively by these hot springs. And so we thought, you know, it'd be nice to drill $10 million boreholes all the way across this entire thing, but there's literally no one who will ever fund me to do that, and also I don't want to get a permit to drill holes all over Costa Rica. So we use these hot springs as like passive sampling ports. And this is the most interdisciplinary collaborative group of people that I possibly work with. And this is not everybody who's involved, but I just want to thank them up front. Originally, these are my lab crew. TJ Rogers is going to turn in his dissertation in a week and a half. He is texting me constantly. He's doing great. He's going to pull through. And then Joyce finished up, and Kate did her master's, and she did wonderful work on this. And then we had all these international collaborators from all over the place, some of whom y'all probably know, including lots of people in Costa Rica, because that is where science has to come from. When you're doing work on, you know, I started out in oceanography, and you just go to oceans and drop things in them because it's all big oceans. But when you're working on land, you do not go in a country that is not your country and take samples, which I'm sure you all know, but it's not just is it morally wrong, but you miss the local knowledge, you miss the understanding of what's actually happening there. So this is driven largely by my colleagues in Costa Rica. So we sampled, this is pretty much the whole country of Costa Rica. So you can see we covered some pretty good ground. This is their line of volcanoes. Volcanoes pop up because this downgoing plate basically runs into mantle, and all the water that on this downgoing plate exsolvates the mantle and makes it sort of have this runaway liquidification that shows up in volcanoes. And our idea was to sample all these red dots, or hot springs, and see if we could get a difference in the progression of the subsurface biosphere as it moves from early subduction into the volcanoes and late subduction. And just to show you what some of the sites looked like, this is Quebrada Naranja. Spanish speakers, I apologize. I'm trying to learn that language and I'm really bad at it. But there was all this iron ferry hydroxides that were produced from these subsurface microbes. So we're sampling gases. There's gases bubbling out of here, but it's at the edge of a river. So we're fighting the surface stuff all the time. Some of the samples look like this, boiling mud pots. So this is looking quite inhospitable for science. Whatever, I don't have to point to that. It's in the picture. And some of them were in the middle of volcanoes. So this is Poas volcano, which is this caldera. You can tell that it erupts a lot. Obviously, because, okay, you see the part with no trees, right? I don't have to laser point to it. Oh, I can point like this. All around this central, so this right here, this eye of it, is basically the throat of the volcano. This is the part where the eruptions start. And you can see that it erupts a lot because the plants would love to use all this good iron, but they can't because every time they get in there, they get blown up. So this is me sampling next to this lake. So this is Donato, he's reading the numbers out to me, and it was 0.85 pH. So it's very, very acidic, basically sulfuric acid. Then you can see the fumes. This is a very uncomfortable picture. The ground is 100 degrees Celsius, and it's really like bad rock. So if you slip, you fall into a battery acid lake. So, but the most dangerous thing about volcanoes is about 54, 56 days after we were in it, this happened. So that you can see the shore where I was. It was like right there. And you can look as it continues erupting. It's not like it just blows up once. You can see this stuff kind of shooting out here again. You should be able to see it. And it's like, if that happened while you were there, there's some more. If that happened while you were there, it's like you would go up and down, then you go up again, then you get a bunch of rocks on your head. It would be very bad. My colleague, one of my worst decision-making colleagues, sent us videos from that crater the day before this. And it was like, the ground was like popping in front of him. I don't know. He's not. I got other colleagues who are a little more circumspect. I worry about that guy. But you know, it's fun to go in these dangerous places, but we also have to mitigate our danger. I don't know. I've got kids. I don't do this stuff. If it had been nasty like that, I would not have gone in any time around this area. I just went in because it was such a calm time. But fieldwork does involve some risks. So the way we think about this subsurface when we get these semi-dangerous samples is we don't know how deep we're getting them from. We are driving all over this country and we are sampling tons and tons of hot springs. They're in people's backyards. It's the coolest thing. We like walk up to people and we're like, hey, and they recognize my colleagues from TV because they talk about volcanoes a lot, and we're like, can we sample your hot springs? And they're like, absolutely. So we sample this stuff and we don't know how deep it's coming from. So one of the things we want to know is, how do we blank out all this crap? Beautiful crap that we depend on, but we don't want to study. So all of the dissolved organic carbon has a carbon isotope. I'm not going to explain what they are. Some of you know intimately about carbon isotopes, and the other 2/3 of you are like, I don't know what this is. But that's okay. Just know that this value ranges, everything up here at the surface ranges from -23 to -30. And our inorganic carbon, so this is carbon dioxide, is like 0-ish. And this should be similar in all springs. So when we measured these things, the DIC, which for atmosphere should be sort of around here-ish, it got quite negative. It got quite light. And it had this pattern with its concentration. And that was sort of intriguing to us. And so my colleagues, who are gas geochemists, looked at this and they said, oh my God, this is a Rayleigh distillation. This is what happens with isotopes. F is the reaction extent, so this is like how far the reaction has progressed. As you have the reaction happening in an open system so that the isotopes can be removed, and there's a fractionation of the isotopes as this progresses, you get this extreme push of the fraction of the del C13 of the values. And so they modeled this stuff and found that actually this was a Rayleigh fractionation that differed between early in subduction, closer to the volcanoes, and in the volcanoes. And these are the volcanic gases themselves. So the offset between this end member and the volcanic gases was basically showed that this stuff was entirely from deep gases. This was not carbon dioxide from the atmosphere at all. So I don't know if you have ever done this with your data sets. You might call it p-hacking, but I don't want to call it that. We don't do that in GL. I swear, that's like just a medical thing. I think we shouldn't limit ourselves. Does anybody see two little rainbows right there? Do you see two? Like, I got this data set and I was like, I swear there's two of them. And I was like, but I shouldn't do that because that's reading too much into my data. But I stared at it for long enough and I finally figured out what was driving those two. If you divide them and you say, okay, this one's on the top curve and I'm making it blue, and that one's on the bottom curve and I'm making it orange, and I tried to figure out, are they different temperatures? Were they different pHs? None of it. Nothing was making sense. And then I put them on a map, and they fall down a line. And I was giving this talk at the Carnegie Institute in DC before we published this paper, and I said, yeah, and we have no idea. For some reason, everything up there is on the upper line and everything down here is on the lower line, but it's real because it's not randomly distributed. And I don't know, maybe there's something out here. And this geophysicist like ran up to me after the talk and he was like, there's a plate boundary that divides those two samples. And he just knew because he knew the plates really well. And so this is the East Pacific Rise and the Cocos-Nazca Spreading Center originating things. So basically, this means that the stuff we're getting coming out of these hot springs, like literally next to toucans and monkeys, like driving around like sipping pina coladas all over Costa Rica, is so connected to this stuff that is subducting offshore that we can see the isotopic difference in the carbon that's coming in from these downgoing slabs, which gives us great power to say that, yeah, this stuff is deep subsurface. This is actually connected to this stuff. So now how do we use this power? We know all this carbon, this inorganic carbon, which is the starting point for chemosynthesis. We know all this stuff is coming from the deep earth. How do we show that the microbes are turning it into life? Or are they just using the plants and cow poop and stuff like that? So now if we put in the organic matter and put it on this same plot, the del C13 of the organic matter, we see it follows along this same line. And it's always offset from what the inorganic carbon were. And the value of that offset is roughly the fractionation that we would expect to see if it was all being made by chemosynthesizers in situ. And so for this reason, we were able to conclude that you can never say all, pretty much everything that's living there seems to be made from the chemicals that are there from the deep earth. So this is despite all the jungles, despite this huge surface signal, we're getting something that's truly in the subsurface, and it's only from there. And we have a positive control, because it's nice to have those. Can we control for jungle farm surface world? And yes, we can. We scooped up sediments where we were across the arc in the same places, and there the carbon is photosynthetic. So we can see the photosynthetic stuff there. So basically, the sort of point in going through all this isotopic stuff is to say that this subsurface of the subduction zone is dominated by chemosynthesis of chemicals. So this is something that could be happening on another planet that has no photosynthesis at all. And now we're at 60 minutes, and I have metagenomes to talk about. But I don't want to wear out my welcome. Do you guys want to, do you know what a metagenome is? Wait, raise your hand if you know what a metagenome is. Oh my God, it's so many people. It's beautiful. Is that like, does it make you want to cry thinking about it? Yeah. Yeah. There's a lot of pain. For those of you who didn't raise your hands, metagenomes are all the DNA in a sample, like all of it. Just sequence it. So what you get is this like garbled mess of every gene from every organism doing God knows what. And so one thing we do with them is that we can knit them back together through the magic of bioinformatics, and I'm not going to tell you how, but I will if you want to know. Not right now. Nobody wants to hear that. But I did not think this could be done at all. But now it can, and it can be done so well that I actually use it. And what this does is it gives you genomes, basically. You can get like a whole genome and say what a thing can be doing. So my student TJ caught 403 of these things that were pretty complete. And so now, think about what you do. All right, you've got like 20 samples across Costa Rica. You know they're something special. They're not a bunch of cow poop. So now you have 400 mags, and you can figure out their distribution because you can recruit those reads. I know some of y'all know what that means, but basically you can see the prevalence of each of those mags in all these data sets. How do you visualize that data? How do you deal with that fire hose? You know, like, so anyway, what we thought of to do, which may not be the only thing, is to make a heat map. I love making heat maps. Just show you like what the color of it shows you how much of it is there. And this is what part of it looks like. This is a third of this heat map. And what this is each column running vertically here is a hot spring. And every row is a microbe. And so if it's yellow here, then that microbe was present in that hot spring. So look at that big black part right there. This is a whole slew of hot springs that did not have those microbes present at all. And so we organized it. I mean, this isn't just randomly distributed. We said put things together that have more similar distributions. And what we were really surprised to see is that it came out to be so completely organized. So this just happens to be all of the volcanic samples came out here. And then all of these sort of like Technicolor things over here are their different metabolisms. So this is just like a ridiculously complex data set visualized as best as we can. So you can kind of see what they're breathing. And I know you can't really see, but this is their respiration and whether they fix carbon, whether they're lithoautotrophs or not. And then this is the same heat map just cut and moved up there. So this is not a separate run. Now, all these ones that were black over here are now yellow because these set of microbes are the ones that are present only in the early subduction. So things filtered out really, really distinctly. And then we had a third group of things that were just kind of everywhere. And so when we looked at what these things were, we wanted to see, are these the ones that are making the energy for everybody else? Yes, they were. We found that about 30% of the community could do carbon fixation, both in this is just same heat map, just cut up into pieces, both in the volcanoes and at the beach, and then less of them were distributed everywhere. And these ones used this oxic carbon fixation pathway everywhere. So we're starting to side eye the ones that are everywhere, as you should be as well. But what is it that distinguished them between these different places? So can you see, it's another test. If you look at the color things, the color strips, do you see differences? Does the left-hand color strip look like different colors than the middle color strip to you and the right-hand color strip? You can kind of see them. This is one reason why I like looking at complex data out like this, because you can really see patterns. Like, for instance, this green line here it's not as complete over here. It's like more of a going concern here. And there's more of this purple stuff here than there is over here. So what this all boils down to, if you stare at it for months and months, is that this volcano stuff liked a little bit of oxygen. They did a lot of sulfur oxidation, and they did nitrate reduction. The things out at the coast were totally different. They were breathing totally different things. They were doing sulfate reduction and hydrogen oxidation. And these guys that were everywhere were aerobic and sulfur oxidizers, and they basically depended on oxygen. So what we concluded from this is that these are the microbial communities that show up late in subduction, because there's a lot of hydrothermal alteration of their waters that adds sulfur, so they have a ton of CO2. There's very little calcite precipitation, and they get lots of oxidized substrates because they have a shallow geothermal gradient that sucks in oxygen. These guys were early in subduction. They didn't have a lot of hydrothermal alteration, but they had lots of reduced sulfur compounds. So they were very carbon dioxide limited, but they still did chemo. They still managed to like fix the carbon and make the ecosystem go, but it was fundamentally different than what we saw at the volcanoes. And then, I don't know, does anybody have any theories for what's going on with these guys who show up literally everywhere? Primary producers, maybe. Oh, like photosynthetic primary producers. If that's what you're implying, then yes, because this is the crap. This is the soil stuff. This is the surface world. The identities of these microbes are like common soil things, and they liked oxygen, and they showed up everywhere. They didn't really care where they were in subduction. So what this means is that the redox couples that fuel this chemosynthetic primary production are distributed, they're everywhere. So if we're gonna go looking somewhere else for this subsurface community, it's likely to be there. As long as we have some sort of energy, we're likely to find it. Conclusion slide. Subsurface life is different than surface life. It seems simplistic, but we know it for sure. They can survive on this is an understatement, very little power and very long time scales. And at least on Earth, we got a whole world down there that is doing its own thing, and it does not need the sun. And they're large enough to really leave a mark on the subsurface landscape. They are precipitating calcite, they're leaving iron ferrohydroxides, we can see them. So I will end with a question for y'all, and I use y'all advisedly. I'm from North Carolina, I live in Tennessee, I'm moving to California, but don't give up on the South. Seriously, I know it's easy to vilify the South. We are gerrymandered all to hell. So we are a fantastic place, and we have very oppressed people, and we deserve your attention. So, question for y'all. If we have this robust of a chemolithoautotrophic, this chemosynthetic subsurface community here, on this place where we have this dominant signal, what will it be like on a planetary body that doesn't have all that stuff at the surface? Will it be different? Will it be the same? And how do we look for it? And I got to thank the funders, and I have a pretty place to work for the next year. And I'll take more questions if you want, or you can do that nap thing.
I was wondering how different the mineralogy was, because I know you had a section that had these hydrogen supported, and I'm assuming that was geologic hydrogen from the serpentinization. Like, how different was the mineralogy? Like, could you tell the difference in what led to in terms of like the type of enzymes that they were able to have in their like biology based on the metals that were around? Like, was that some kind of connection that you could make, or? I don't know, I just want more elaboration, I'm sorry.
Yeah, I want more elaboration on the minerals too. Shauna Morenson did all the mineralogy for this, and she found pyrite framboids in the very altered hydrothermal. So that's feeding into our hypothesis of why there's so much iron and sulfur cycling in the volcanoes. But picking apart how the metals influence and the crystalline form of the metals influence all these communities is a bit of a fine scale thing, and so I think that's going to be more apparent in our global data set because this project has metastasized.
So my question is coming from very limited background in biology. So
Sweet.
yeah, so when you talk about lifespan, why do different organisms have different lifespans? Like different species have different lifespans. Do we know this?
I don't know. Why do different species have different lifespans?
Yeah.
Yeah, that's a great question. Somebody want to answer that?
Kelly, do you want to yell down for us?
Yeah, Kelly, tell us. Oh, there's a question. I don't think they're feeding the rest of the communities. Oh, you don't? No, I think they're just really angry to have been washed into a hot spring. I don't want to use the word contamination because that kind of means, that implies something from our bodies or like our coughs. It's more like just not the things we were looking for. Because a hot spring is coming up through layers of rock. And so there's going to be some soil stuff washed in. Yeah, they could be. Did you have a thought? I mean, they could be feeding them.
No, my understanding was that you guys were thinking that these were, what was, like, producing things that were feeding the others that were there?
I think the chemosynthesizers are. I mean, they're basically taking carbon that's coming from that slab. So that's carbon that got buried in the Pacific Ocean 10 million years ago. And it gets buried and gets volatilized, however it gets from high pressure and temperature, and then comes back up. And that's what's forming their bodies. Claire?
Thank you for your talk. I was wondering, so you talk a lot about the unculturable. That's what you work on.
Actually, never say that word.
Okay, I'm sorry.
I get in trouble when I do.
So a lot of these organisms, or most of them, are
Uncultured.
Uncultured.
Yeah.
So what are your thoughts on you are annotating a lot of this stuff from stuff that is also uncultured? Do you think there is just like that hypothetical protein sort of idea where this is most related to this, which is most related to this? How do you think you can sort of circumvent that potential issue?
You mean the issue of the game of telephone where you think something's a one thing and it's actually not?
Right.
I think that we have to be really careful about what we conclude. Like, if you're, at least in my research, like if my story really depends on a particular annotation, then I go hard into that annotation and make a multiple species alignment multiple sequence alignment of that gene and show that there's more evidence for it being what I think it is than just like Proca annotates it as X. And you can look, you can look for the PFAMs, you can look for the active site, you can do all sorts of things to sort of prove to yourself. You can look for the flanking genes, the gene synteny, all these things you can use to build up a case. You're not going to do that for a whole genome, or much less 400 genomes, but you can do that for the thing that you want to talk about. But I just think that like there's so much to be gained in that hypothetical space. And sorry, another soapbox about this is that, cool, we free ourselves from cultures. We're free. What's the first thing we do when we get this genome? Say, how similar is it to a culture? So we're not free.
Great talk. I should preface this by saying that I'm an astronomer, so I did my best.
Well, the last one just stumped me, so.
Mars and Europa and Enceladus and those icy moons have very different environmental conditions than the Earth.
Yeah.
And it seemed like a lot of this was predicated on subduction
Yeah, I know all of this.
not some sort of
Stagnant
subsurface
lid
ocean
tectonics,
world.
for instance.
Yeah. So, I just wonder how you develop a subsurface biological world in a static lid environment or in a subsurface ocean environment.
I'm writing an exo grant about that right now, actually. But I mean, not that I know the answer to it. But I mean, that is basically like the fatal flaw of my second half of my talk. But it's a relevant one because there's no plate tectonics in our solar system except on Earth. In the entire history, the fact that Olympus Mons is so enormous is simply because that hotspot's been sitting there like for however long it's been sitting there. So I think that the grant that I'm writing right now is, we may not get it, is to basically try to get out of subduction zones and do our same style of sampling and our same style of work around hotspots here on Earth, or at least plume-influenced environments. But you can't get away from plate tectonics on Earth just the same way you can't get away from the photosynthetes. So we're going to have to deal with Hawaii.
So are you suggesting there's no life elsewhere?
No. Can you imagine if that was the conclusion of my talk? Like, everybody go home. That would be awesome. I love that. No. I think the answer to that is going to be fascinating.
I guess this is kind of to that kind of effect. So, is there something like, why do we have plate tectonics?
Oh,
Sorry, this is
somebody-
almost
no,
for the question asker.
It's a great question. I don't know why. Do people know that? I feel like people know that. Why do we have plate tectonics? I don't know. It maybe has something to do with our size, our size and relative heat. I mean, because it's all heat convection that's driving it. So you need the right brittleness versus heat to break it apart and move them, but that may not be the right answer. I don't know. I would say I'm a microbiologist, but I don't have any degrees in that. Did you? I think you raised your hand. You raised your hand first, didn't you? I think you were like the first person.
Is this on? Whoa. All right, let's see if this makes sense. So, hi, I'm Emily. I study the limits of life in saline environments.
Nice.
And there's some research on thermodynamic limits to certain metabolisms, including autotrophy that's not driven by oxygenic things. And so I was wondering, given that you found a lot of chemoautotrophs in the deep subsurface that are in incredibly energy-limited environments, do you think there actually is a limit to life? Or is this, I don't know.
You mean an energetic limit to life?
Yeah, sure. In an extreme environment, you have to use a lot of energy to survive there, right?
Yeah.
So, yeah.
Even places where you get your protons for free, like an acidic environment, you still spend a lot of energy bailing.
Yeah, sure.
Is there a limit for life? Yes. Yeah, there has to be. Like a Gibbs free energy of 0 is equilibrium. And you can't have life with equilibrium. There has to be a gradient. But the interesting question is, where is that line? And that's what we don't know. But there is. It exists. Did you want to ask your question?
Hi, I'm also an astronomer, and I wanted to kind of move even further out than Nick did. So something we think about a lot as astronomers is habitable zone and what temperature planets might have to be to host life. But I'm wondering what your thoughts are about the prospects for life on free floating planets that aren't associated with a star.
There are free floating planets that are not associated with stars?
Yes.
Seriously? Is this like a Kepler finding? There's a lot.
It's more of a microlensing.
Do we know about them from
thing.
Kepler, or what do we know about them from even Hubble? Okay.
I have a lot of thoughts about this. I'd be happy to talk about it sometime.
That's crazy. Wait, but how do they have? Do they have their own heat source internally? Or they? Because you got to have some kind of gradient. That's all. You got to have something.
Like some heat from formation, probably.
Yeah,
that's gradually
right.
fading over time.
Or some tidal forcing from somebody nearby, or are they just totally alone? They got nothing. Wow. The radiogenic decay. Yeah, yeah. So you got to have something, right? There's got to be a gradient. There's got to be, if not redox, just something. I don't know. Clearly, I don't know the answer to that because I didn't know that they were there. But we talk about the habitable. When you think of the habitable zone, do you think about distance from the star and brightness from the star Also, think about the depth of that planet, because that'll really expand your habitable zone quite a bit. If you can go inside the planet, it gets better.
We have time for one more question.
Free floating planets.
Hi. First off, I'm also from North Carolina, so hello.
Where are you from?
Raleigh.
Cute. Sweet. I'm from Beaufort.
Oh, nice.
I went to Science
Anyway,
and Math.
I was just curious, what do you think is the stability of these subsurface ecologies through both geological time and planetary scales? And if there's any way to have a fossilized record of their presence at all that we could detect?
Yeah, the carbonates, the minerals that they leave behind, the waste products, are probably the best. And microfossils, of course, and lipids are good ways to look for these sorts of things. But in terms of the time span of this stuff, we're just looking at the modern stuff now. And so I think that I don't know as much as I should about that, but there's a lot to be done.
All right, well, let's give one last round of applause for Karen. Just a few housekeeping things. So tomorrow morning, we'll have breakfast at the grad student lounge. So those of you who didn't come, it's a few doors down. If you look at the FAQ we sent out, all the information's in there. If there's any questions that are not answered, just message Miguel or I. But that will start at 8 a.m. and then we'll promptly start this talks right at 9. Great. Oh yeah. And
okay, I just want to add one thing. For those of you that checked in and got your awesome swag bag, that's great. But if you did not get the plastic NASA bag and you want one, come see me at the check-in desk before you go, okay, guys? Thank you.